Hey there, curious minds. Welcome to another deep dive. Today we're taking quite a stack of information, actually a whole chapter from Essentials of Genetics, 10th edition. And we're going to try and distill it into, the most important, mind-blowing nuggets. Our mission, to unpack the incredible world of recombinant DNA technology, from its, surprising origins right up to its cutting-edge applications today. You've probably heard terms like gene splicing or maybe genetic engineering. But what does it really mean to, stitch together DNA from totally different sources? How did scientists even start thinking about this? And maybe most importantly, what does it mean for you? Whether you're prepping for a meeting, catching up on the field, or just, super curious about how life works.
It is pretty astonishing, isn't it? I mean, this technology, it earned Nathan, Smith, and Arbour the Nobel Prize back in 1978, and rightly so. It just completely transformed our ability to isolate, replicate, and really analyze specific bits of DNA. What's so fascinating, I think, is the leap in thinking. Once you could grab hold of a single gene, you could then make millions of identical copies, clones, right? And then start manipulating them for research, for commercial products, you name it. This work, it really laid the groundwork for, the entire field of genomics. It lets us read and even rewrite the instruction manual of life.
Okay, let's definitely unpack that. If we're talking cutting and pasting DNA, we need some serious tools. What were the molecular scissors in the delivery trucks that made it happen?
Right. The initial breakthroughs really depended on two key tools, restriction enzymes and cloning vectors. So first, those molecular scissors, the restriction enzymes, these aren't something humans invented. They're actually made naturally by bacteria. It's a defense mechanism they have against viruses. Yeah, they restrict the virus by chopping up its foreign DNA. Scientists have found, over 4,300 of them. Hundreds are used in labs now. And the amazing part is their precision. They recognize and bind to very specific DNA sequences.
Called restriction sites.
Exactly, restriction sites. And then they cut both DNA strands right there within that specific sequence. Very precise cuts.
And I remember reading something interesting here. These recognition sequences, they're palindromic. They read the same forwards and backwards?
That's it, palindromic. So 5' to 3', it reads the same on both strands. Think GATC on one, CTTAG on the other.
Okay.
And this leads to two main types of cuts. First, you have cohesive ends or sticky ends, like the ones E. coli makes. The cuts are offset, leaving these little single-stranded overhangs.
Like little sticky bits ready to connect.
Precisely. And they can easily base pair with any other DNA cut by the same enzyme, no matter where it came from. That's the key. Then you also have enzymes that make blunt end fragments. They cut straight across, both strands at the same point, no overhangs.
Okay, so you've used your enzyme, you've made your cut, but the pieces are still separate. What sticks them back together? What's the molecular glue?
That would be DNA ligase. It's another enzyme, and its job is to form those strong phosphodiester bonds. It covalently joins the fragments, sealing the gaps and creating your brand new recombinant DNA molecule. It really is the molecular superglue here.
Okay, scissors, restriction enzymes, glue, DNA ligase. Now, what about those delivery systems you mentioned, the cloning vectors?
Right, the cloning vectors. These are basically DNA molecules that act as carriers. They accept the DNA fragment you want to clone, and then they can replicate themselves inside a host cell, usually bacteria. They need a few key features, like multiple restriction sites, so you have choices for inserting your DNA. They need to replicate independently from the host's own chromosome. And crucially, they need a selectable marker gene, often something like antibiotic resistance.
Like AMPAR for ampicillin.
Exactly, like AMPAR. Or sometimes a reporter gene, something like LAC-Z, that gives you a visible signal, like a color change, to show if the cloning worked.
And when we talk vectors, the ones you hear about most, especially early on, are plasmids, right?
Yes, modified bacterial plasmids were definitely among the first and are still workhorses. There are these naturally occurring small circular bits of DNA found in bacteria separate from the main chromosome. Getting them into the bacteria, that process is called transformation. You can use calcium ions and a quick heat shock.
Right.
Or a method called electroporation, which uses a jolt of electricity to make temporary holes in the cell membrane. Once inside, the bacteria treat the plasmid like its own DNA and replicate it along with their own, making hundreds of copies very quickly.
You mentioned reporter genes, like LAC-Z.
Yeah.
How does that help you know if your specific DNA fragment actually got into the plasmid? Because sometimes the plasmid might just close back up, right?
That's a really important practical point. A very clever strategy is blue-white screening using that LAC-C gene. So the LAC-C gene codes for an enzyme, beta-galactosidase.
Mm-hmm.
If you put a chemical called XGAL on the growth plate, colonies with a working LAC-C gene turn blue. Now, if you design your plasmid, so the place you insert your foreign DNA, the multiple cloning site, is right inside the LAC-Z gene, then successfully inserting your DNA fragment will actually break the LAC-Z gene. It won't make the functional enzyme anymore.
So if the gene is broken, no blue color.
Exactly. So when you grow the bacteria on plates with the antibiotic to kill cells without any plasmid and XGAL, colonies with the original empty plasmid will be blue, but colonies that contain your recombinant plasmid, the one with your DNA inserted, will be white.
That's brilliant. A simple visual check. White means success.
It's a very effective screening method.
But plasmids, they're great, but they have a size limit, don't they? Like, you can't stuff a really huge piece of DNA into them. Maybe 25 kilobases, max?
That's right. They do have limitations on insert size.
Yeah.
So for larger DNA fragments, scientists needed other tools. They developed phage vectors using modified bacteriophages, like phage lambda. These can carry bigger inserts, up to maybe 45 kilobases. They infect bacteria, and instead of colonies, you look for clear spots called plaques.
Okay, bigger capacity. What about even larger pieces?
For really large fragments, they developed bacterial artificial chromosomes, or BACs. These are essentially very large plasmids, capable of holding 100 to 300 kilobases, huge chunks. And for the truly massive pieces, we needed yeast artificial chromosomes, YACs. These actually mimic eukaryotic chromosomes. They have telomeres, origins of replication, even a centromere. YACs can handle inserts from 100 up to 1,000 kilobases, a million base pairs.
Wow.
They were absolutely essential for projects like the Human Genome Project, for mapping and sequencing those enormous stretches of DNA.
Okay, so BACs and YACs for the big stuff. You also mentioned expression vectors earlier. How are them different?
Right, that's an important distinction. All the vectors we've discussed so far, plasmids, phages, BACs, YACs, they're primarily cloning vectors. Their main job is just to carry and replicate the DNA insert. Expression vectors, on the other hand, are specifically designed to do more. They contain the necessary signals promoters, ribosome binding sites, terminators, to ensure that the gene inserted into them is actually transcribed into mRNA and then translated into protein within the host cell.
So they make the gene do something, produce its protein product.
Precisely. This is absolutely vital if you want to study the function of the protein, or if you're in biotechnology and you want to use bacteria or yeast as little factories to produce large quantities of a valuable protein, like insulin or growth hormone.
Okay, that makes sense. So we've got cutting, pasting, replicating, even expressing genes. Now imagine you've created this huge collection of cloned DNA segments, like a library. How do you find the specific gene, the one book you're looking for, in that massive collection?
That analogy is spot on. We call them DNA libraries, collections of cloned DNA fragments, and there are two main kinds. First, genomic DNA libraries. These aim to contain overlapping fragments that represent the entire genome of an organism. So that includes all the coding genes, but also all the non-coding regions, the regulatory sequences, everything. It's the whole book.
Got it. The complete set.
Then you have complementary DNA libraries, or cDNA libraries. These are quite different and very useful. They are made starting from mRNA molecules isolated from cells. Remember, mRNA only represents the genes that are actively being expressed or turned on in those cells at that specific time.
So it's not the whole genome, just the active parts, like a snapshot of what the cell is doing right now.
Exactly. It's incredibly powerful. If you want to know which genes are active in, say, a cancer cell compared to a normal cell, or during embryonic development, or in brain tissue versus liver tissue, you'd make a cDNA library from those specific cells.
That's a really key distinction. It tells you about gene activity.
Precisely. Very valuable for studying development, disease, how cells differentiate. Creating cDNA involves an enzyme called reverse transcriptase. It does the opposite of normal transcription. It uses RNA as a template to make a complementary DNA strand.
Okay. And how did they used to find genes in these libraries before we could just sequence everything?
Historically, they used a technique called library screening, often using a probe. A probe is a labeled piece of DNA or RNA that has a sequence complementary to the gene you're searching for. You'd essentially use this probe to find and stick to the clone containing your target gene within the library. While whole genome sequencing has kind of replaced this for just finding genes now, the library concept, especially cDNA libraries, is still vital for understanding gene expression patterns.
Right. Now, speaking of things that completely changed the game, Polymerase chain reaction, PCR. It sounds like this just bypassed the whole need for cloning in bacteria sometimes. Amplify DNA directly. Kary Mullis won a Nobel for this one, too.
Oh, absolutely. PCR was and still is incredibly revolutionary. Its power lies in its speed and sensitivity. It lets you amplify a specific target DNA sequence exponentially, in vitro, right there in a test tube. And you can start with minuscule amounts of DNA, like tiny traces from a crime scene, dried blood, a single hair follicle, or even DNA from ancient fossils.
That's amazing. How does it actually work? It's a chain reaction, so what are the steps that repeat?
It's a cycle with 3 main steps, repeated over and over in an automated machine called a thermocycler. Step 1 is denaturation. You heat the DNA sample, usually to around 92-95 degrees Celsius. This melts the double helix, separating it into 2 single strands.
Okay, strands separated.
Step 2 is hybridization or annealing. You lower the temperature, typically somewhere between 45 and 65 degrees Celsius. This allows short, single-stranded DNA pieces called primers to bind or anneal to their complementary sequences on the template strands. You need 2 primers, one for each end of the target sequence you want to amplify.
Got it. Primers bookend the target.
Exactly. And step 3 is extension. You raise the temperature slightly, usually to around 65-75 degrees Celsius. This is the optimal temperature for the DNA polymerase enzyme to work. It latches onto the primers and starts synthesizing new DNA strands, using the original strands as templates, extending outwards from the primers.
And the polymerase used is special, right? It has to survive the heat.
Absolutely critical. The breakthrough here was using Taq polymerase. It comes from a bacterium, Thermus aquaticus, that lives in those boiling hot springs in Yellowstone. So Taq polymerase is thermostable. It doesn't get destroyed by the high denaturation temperatures. It just keeps working cycle after cycle.
Wow. And each cycle doubles the DNA.
Essentially, yes. Each cycle doubles the amount of the target sequence. So if you run, say, 25 or 30 cycles, which only takes a few hours, you can get a millionfold or even billionfold amplification of your target DNA. Incredible amplification from almost nothing.
That sensitivity is just wild.
Yeah.
So beyond making lots of DNA copies, what are the big real-world applications of PCR?
Oh, it's everywhere. It's a cornerstone of diagnostics. Detecting infectious agents like bacteria and viruses, HIV, hepatitis, COVID-19, you name it. Forensics, identifying individuals from trace DNA. Basic research, amplifying genes for sequencing or cloning. And there are important variations too, like reverse transcription PCR, RT-PCR. Here, you start with RNA, use reverse transcriptase to make cDNA, and then amplify it by PCR. This lets you measure gene expression levels, how much mRNA is present.
So you can see how active a gene is.
Exactly. And then there's quantitative real-time PCR, qPCR. This lets you measure the amount of DNA being amplified during the reaction in real time. It gives you very precise quantification.
Powerful stuff. Are there any downsides or limitations?
Well, the main ones are you need to know some sequence information about your target to design the primers. You can't just amplify something completely unknown. And because it's so incredibly sensitive, contamination is a major concern. Even a tiny amount of stray DNA can get amplified and give you a false result if you're not careful.
Right, meticulous lab work needed. Okay, so we can clone DNA, we can amplify it with PCR. How do we actually look at these DNA fragments, see their sizes, maybe identify them?
The absolute workhorse technique for that is agarose gel electrophoresis. It's routine in almost every molecular biology lab. You prepare a gel, usually made of agarose, which forms a kind of porous matrix. You load your DNA samples into little wells at one end, then you apply an electric current. Since DNA has a negative charge because of the phosphate groups, it migrates towards the positive electrode.
And smaller pieces move faster.
Exactly. The gel matrix acts like a sieve. Smaller DNA fragments wiggle through the pores more easily and travel further down the gel in a given time. Larger fragments get tangled up more and move slower. So it separates the DNA fragments purely by size.
How do you see them then? DNA is invisible.
Right. You stain the gel with a dye that binds to DNA and fluoresces under UV light. The most common one historically was ethidium bromide, though safer alternatives used now too. Under UV light, you see bands, each band representing a collection of DNA fragments of the same size. You can estimate the sizes by comparing them to a ladder, a sample containing DNA fragments of known sizes run alongside your samples.
Okay, so gels let you see and size fragments. What about those blotting techniques? Southern, northern, western? Sounds like a compass direction thing.
It does. Southern blotting came first, named after its inventor, Edwin Southern. It was a really groundbreaking technique. You'd run your DNA fragments on a gel, just like we discussed, but then you'd transfer the separated DNA fragments out of the fragile gel onto a more robust membrane, like nitrocellulose or nylon.
Like blotting the DNA pattern onto a piece of paper?
Kind of, yeah. Then, on that membrane, you'd use a labeled probe again, a piece of DNA or RNA complementary to the specific sequence you're interested in. The probe would hybridize, or stick, only to the band containing your target sequence. So even if your gel just showed a smear of many fragments, the southern blot would light up only the specific band you were looking for.
Ah, so it combines separation by size with identification by sequence. Very clever.
Hugely important for identifying specific genes within a complex mixture, like total genomic DNA.
And that led to the others, northern and western.
Exactly. Building on the same principle, northern blotting is for detecting specific RNA molecules separated on a gel. Great for studying gene expression by looking at mRNA levels. And Western blotting is for detecting specific proteins after they've been separated, usually by a different type of gel electrophoresis, STS page age, and transferred to a membrane. You use antibodies as probes in Westerns.
So it's a whole family of techniques for different molecules.
Yeah.
DNA, RNA, protein.
Precisely. Shows how these molecular biology methods build on each other. And there's another related technique worth mentioning. Fluorescence in situ hybridization, or FA. Here, you use fluorescently labeled probes, but you apply them directly to chromosomes or RNA within cells or tissues, usually on a microscope slide. You don't need the gel or the blotting step.
So you can see where the gene or RNA is located right inside the cell.
Yes. It's very powerful for developmental studies to see which cells are expressing a gene, or in diagnostics. For example, you can use multiple colored probes for different chromosomes to create a spectral karyotype, essentially painting each chromosome a different color. This makes it much easier to spot abnormalities like translocations or deletions, which are common in cancer cells.
Okay, visualizing and identifying is one thing, but to really understand DNA, you need the ultimate detail, the exact sequence of A's, T's, C's, and G's. How did we first crack that code?
The classic method, the one that really enabled large-scale sequencing for years, was Sanger sequencing, also called dideoxy chain termination sequencing, developed by Fred Sanger, who actually won 2 Nobel Prizes.
Wow. How did it work, chain termination?
Yeah, the key was using modified nucleotides called dideoxynucleotides, or DDNTPs. Normal nucleotides, DNTPs, have a hydroxyl group, OH, at the 3' carbon position, which is needed to add the next nucleotide in the chain. But DDNTPs lack this 3' hydroxyl group. So if a DDNTP gets incorporated into a growing DNA strand during replication...
It's a dead end. The chain stops growing.
Exactly. Chain termination. So you'd set up 4 separate reactions. Each reaction had normal DNTPs, DNA polymerase primers, and the template DNA you wanted to sequence. But each reaction also contained a small amount of one specific DDNTP in one tube, DDTTP in another, and so on. As DNA synthesis occurred, occasionally a DDNTP would be incorporated instead of the normal DNTP, stopping the chain at that specific base.
So you'd end up with fragments of different lengths, each ending with a specific known base.
Precisely. You'd then separate the fragments from all 4 reactions by size using gel electrophoresis. By reading which base terminated the fragment at each length, you could deduce the DNA sequence.
That sounds painstaking. How did they improve it?
The big improvement was using fluorescent dyes. Instead of 4 separate reactions, they labeled each of the 4 DDNTPs, DDA, DDT, DDC, DDG, with a different colored fluorescent dye. Now you could run everything in a single reaction tube or lane. The fragments were separated by size using super thin capillary gel electrophoresis. As the fragments passed a laser beam, their fluorescent color was detected, indicating which base terminated the fragment. This generated a computer readout called an electropherogram or chromatogram, showing the sequence as colored peaks. Much faster and automated.
Okay, Sanger sequencing, the classic. But you used the word historically. What's taken over now? Genomics seems to have exploded.
It really has. The demand for faster, cheaper sequencing, driven by things like the Human Genome Project and personalized medicine, led to a revolution. We entered the era of next-generation sequencing, or NGS. These technologies are vastly different from Sanger. They use massively parallel approaches. Instead of sequencing one DNA fragment at a time, they sequence millions or even billions of fragments simultaneously on a tiny chip or flow cell.
Millions at once.
Different platforms use different chemistries, but a common principle is sequencing by synthesis. Tiny DNA fragments are attached to a surface, amplified in clusters, and then sequenced as polymerase adds labeled nucleotides, with detectors recording the signal from each cluster in real time. Platforms like the Illumina systems can generate enormous amounts of data terabases very quickly and at a dramatically lower cost compared to Sanger, enough to sequence multiple human genomes in just days.
Incredible scale and speed. Is that the cutting edge, or is there something even newer?
The current cutting edge is often called third-generation sequencing, TGS, sometimes long-read sequencing. The big difference here is that these technologies aim to sequence a single molecule of DNA in real time without needing to amplify it first.
A single molecule. Wow.
Yeah. For instance, Pacific Biosciences, PacBio technology, uses special structures called ZMWs, zero-mode waveguides, to watch a single DNA polymerase molecule incorporating fluorescently labeled nucleotides as it copies a single DNA strand. Oxford Nanopore Technologies, ONT, uses protein nanopores embedded in a membrane. As a single DNA strand is fed through the pore, it disrupts an electrical current in a characteristic way for each base, allowing the sequence to be read directly. They even have portable USB stick-sized sequencers like the Minion.
A USB sequencer. That's science fiction stuff.
It feels like it. Now, the accuracy of these TGS methods was initially lower than NGS, but it's improving rapidly, and their huge advantage is generating very long reads, tens of thousands, even hundreds of thousands of base pairs from a single molecule.
Why are long reads so useful?
They make it much easier to assemble complex genomes, especially regions with lots of repetitive sequences that confuse shorter reads. They can span tricky regions and help resolve ambiguities.
Okay, so sequencing tech is just racing ahead. Now, shifting gears a bit, we've talked about manipulating DNA in tubes in bacteria. What about changing genes within a living organism to figure out what a gene actually does in the context of a whole animal?
Right, that's a crucial step for understanding gene function. This involves gene targeting approaches, leading to things like knockout animals and transgenic animals. Let's start with knockout animals, often KO mice. In these animals, a specific gene has been intentionally inactivated or, well, knocked out. It's a loss-of-function approach.
So you're basically asking what happens if this gene isn't working?
Exactly. You create the knockout, and then you carefully observe the animal's phenotype, its physical traits, its behavior, its biochemistry. Any differences compared to a normal animal can tell you about the function of the gene you eliminated. This work, pioneered in mice by Kopecky, Smithies, and Evans, another Nobel Prize, has been fundamental for creating animal models of human diseases.
How do they actually make a knockout mouse? It sounds complicated.
The traditional method is quite labor-intensive, yes. It involves creating a targeting vector in the lab which contains a mutated or disrupted copy of the target gene. You introduce this vector into embryonic stem, ES cells, cells from an early embryo that can develop into any cell type. You select the ES cells where the targeting vector has correctly replaced the normal gene through homologous recombination. Then you inject these modified ES cells into an early mouse embryo, a blastocyst, implant that embryo into a surrogate mother mouse. The resulting pups will be chimeras, a mix of cells. You then have to breed these chimeras carefully to eventually get mice that are homozygous null, meaning both copies of the gene are knocked out.
That is involved. What if the gene is essential for the embryo to even develop?
Good point. If knocking out the gene causes embryonic lethality, the mice don't survive. In those cases, researchers often use conditional knockouts. These are more sophisticated systems where the gene is disrupted only in specific tissues or only after the animal reaches a certain age, often using inducible systems like CRISPR.
Okay, so knockouts are about removing function. What about the opposite? Adding or boosting a gene?
That's where transgenic animals come in. These are often called knock-in animals. In this case, you're introducing a new gene, a transgene, or overexpressing an existing gene.
So turning genes on or adding new ones instead of turning them off.
Precisely. You introduce the transgene, often with specific regulatory sequences to control where and when it's expressed, into the animal's genome. This has been used to create, for example, humanized mice that carry human genes, making them better models for testing drugs intended for humans. It's also used in biotechnology, creating transgenic animals that produce valuable pharmaceutical proteins in their milk, for instance.
Knockouts, transgenics, powerful tools. But that knockout process sounded really long and complex. Is there a faster, maybe more efficient way to precisely edit genes inside living cells of organisms now?
Absolutely, and that's where genome editing comes in. The technology that has completely taken over this field is the CRISPR-Cas system. It's faster, cheaper, easier, and often more precise than the older methods.
Ah, CRISPR. You hear about this everywhere. And this also came from bacteria fighting viruses, right? That seems to be a theme.
It is. Another amazing amazing example of basic research on bacterial immunity leading to revolutionary technology. The most commonly used system is CRISPR-Cas9. Cas9 is an enzyme, a nucleus, that acts like those molecular scissors again, making a double-strand break in DNA.
But super targeted this time.
Extremely targeted. Cas9 itself doesn't know where to cut. It needs a guide. That's the single guide RNA, sgRNA. The sgRNA is a short RNA molecule that you design in the lab to be complementary to a specific 20-nucleotide target sequence in the genome you want to edit. This sgRNA literally guides the Cas9 enzyme to that precise spot. Cas9 also needs to recognize a short adjacent sequence called the PAM sequence, usually NG for Cas9, to actually make the cut.
Okay, so the guide RNA finds the address, PAM confirms it's okay to cut, and Cas9 makes the break. Then what?
Once Cas9 makes that double-strand break, the cell's natural DNA repair machinery kicks in. And there are two main pathways it uses. The first is a non-homologous end joining, NHEJ. This pathway is quick and dirty. It just tries to stick the broken ends back together as fast as possible, but it often makes mistakes, introducing small insertions or deletions, we call them indels, right at the cut site.
And those little errors can mess up the gene.
Often, yes. If the indel causes a frameshift mutation within the coding region of a gene, it usually results in a non-functional protein. So NHEJ is actually a great way to knock out a gene using CRISPR much faster than the traditional ES cell method. The second repair pathway is homology-directed repair, HDR. This one is much more precise. HDR uses an undamaged homologous chromosome as a template to accurately repair the break.
Okay, more accurate repair.
Yes, and scientists can hijack this pathway. If you provide the cell with an artificial DNA donor template that has homology to the region around the cut site, but also contains your desired edit, maybe a specific base change, a deletion, or even an insertion of new sequence, the HDR pathway can use your template to repair the break, incorporating your desired change precisely into the genome.
So NHEJ for breaking genes, HDR for precisely fixing or changing them. That's incredibly versatile. What are the catches? Any limitations or concerns?
The biggest concern, especially for therapeutic applications, has been off-target edits. Sometimes the guide RNA might bind to sequences elsewhere in the genome that are similar but not identical to the intended target. If Cas9 cuts at these unintended sites, it could cause harmful mutations.
Right. You want the edits to be only where you intend them.
Exactly. So a huge amount of research has gone into improving specificity. This includes engineering Cas9 variants that are more accurate, developing better computational tools to predict and design guide RNAs with fewer off-target risks, and exploring alternative CRISPR systems beyond Cas9. Specificity is constantly improving.
Okay, so assuming we can manage the off-target issue, what does this CRISPR revolution mean? What are the applications we're seeing now and what's coming?
The applications are incredibly broad. In basic research, it's just accelerated everything. Scientists can now generate knockout cell lines or animal models in weeks or months instead of years to rapidly figure out gene functions. In biotechnology and agriculture, it's huge. Creating genetically modified organisms is much faster and cheaper. We mentioned the virus-resistant pigs.
Yeah, saving millions.
Right. And in food crops, modifying them for better nutrition, longer shelf life, pest resistance, drought tolerance, like corn that needs less water or mushrooms that don't brown. These are already developed, and some are getting close to market. You might see CRISPR-edited foods in grocery stores relatively soon.
Wow. And what about the really big one, medicine, treating human diseases?
That's definitely the area with the most excitement and also the most caution. Gene therapy using CRISPR is advancing rapidly. The idea is to directly correct the genetic mutations that cause inherited diseases. Clinical trials are already underway, or starting, for a range of conditions. Things like sickle cell anemia and beta thalassemia by editing blood stem cells, certain types of inherited blindness, some liver diseases, muscle disorders, even targeting cancer cells or fighting infections like HIV.
So potentially curative therapies for diseases that were previously untreatable.
That's the hope. It's still early days for many of these therapies, and safety is paramount, but the potential is truly immense.
What an absolutely incredible journey we've just taken through this world of recombinant DNA. I mean, from finding molecular scissors in bacteria to building DNA libraries, copying DNA in a tube, and now precisely editing genomes with CRISPR. It's staggering how geneticists can study, manipulate, and really transform life at this fundamental level.
It really is. And, the development of these methods, especially something as powerful and accessible as CRISPR-Cas, immediately brings up profound ethical considerations. Scientists themselves recognized this very early on. Even back with the first recombinant DNA techniques, there was a famous meeting at Asilomar where scientists imposed a temporary moratorium on certain experiments until safety guidelines could be developed.
Right, taking responsibility for the power they were unlocking.
Exactly. And programs like ELSI, the Ethical, Legal, and Social Implications Research Program, funded alongside the Human Genome Project, were set up specifically to anticipate and address these issues. Things like genetic privacy, discrimination, and setting boundaries. For a long time, there was broad consensus against things like germline editing changes that will be passed down to future generations or enhancement therapies purely to make people better than normal.
But CRISPR makes these things technically much more feasible now, doesn't it? Which forces us to revisit those boundaries.
It absolutely does. The power is immense. We could potentially eliminate devastating genetic diseases like Huntington's or cystic fibrosis from families forever. We could potentially engineer mosquitoes so they can't transmit malaria, saving millions of lives.
But the flip side is, where do we draw the line? How do you, listening now, think we should navigate this? Do we have the ethical right, or even the wisdom, to alter the genetic makeup of future human generations? How do we ensure these tools are used equitably, safely, and responsibly for genuine therapeutic needs, and not for, well, frivolous or potentially dangerous enhancements? These are huge questions for all of society.
They really are. There are no easy answers.
Well, thank you for joining us on this deep dive into the truly fascinating and sometimes ethically challenging world of recombinant DNA. We really hope you're leaving with a clearer picture of how genetics is fundamentally shaping our world and the kinds of questions we all need to think about. Until next time, keep digging deeper.